Cell division is a fundamental process, but at the same time it is also high risk. Cells cannot divide at random. The process must occur at the correct time and only under safe conditions. To manage this, cells follow a strict sequence of steps called the cell cycle, which organizes growth, DNA replication, and division. This regulation ensures that genetic information is preserved and passed on accurately. Cells divide for three primary reasons: growth, tissue repair, and reproduction. In multicellular organisms, division allows for development and the replacement of dead or damaged cells, as seen both here and here. It is important to note that cells do not divide on their own. They must receive specific signals from their environment to trigger the process, such as from a different neuron. This ensures that division only happens when it actually benefits the organism and doesn't end up harming it instead. The cell cycle is an organized process with three main parts: interphase, mitosis, and cytokinesis. Interphase is the longest stage, around 90% of the entire time, where the cell grows and prepares for division. Mitosis is the division of the nucleus itself, and cytokinesis is the physical splitting of the cell. This structure ensures that the cell has all the necessary tools ready before the actual division begins and prevents premature division. Interphase consists of three stages: G1, S, and G2. In G1, the cell grows and performs its normal functions. At this point, some cells may temporarily or even permanently exit the cycle into a non-dividing state called G0. Here, they continue functioning, but they do not prepare to divide. In the S phase, the cell copies its DNA, turning every single chromosome into a pair of matching sister pieces joined at the center. In G2, the cell performs a final check and creates extra parts. This ensures that the two new daughter cells will be fully functional. Once the DNA is copied and the cell has reached a proper size, it must solve a mechanical challenge: moving two full sets of DNA without causing any damage at all. This transition marks the end of the growth phase and the beginning of the physical work of division, leading directly into mitosis. At this point, the cell is fully committed to completing division unless a real serious problem is detected. Mitosis is specifically the division of the cell nucleus. The goal is to deliver a complete set of DNA to each new cell. To maintain order, mitosis follows four stages: prophase, metaphase, anaphase, and telophase, often abbreviated as PMAT. This precise sequence ensures that each daughter cell is a perfect genetic copy of the parent cell. The first stage is prophase. During this step, the DNA condenses into tight chromosomes so that the genetic material does not break while it is being moved. The nuclear envelope breaks down to allow movement within the cell. Meanwhile, the spindle, a framework made of tiny tubes, begins to form to help move and organize the chromosomes for the next step. In metaphase, the spindle fibers pull the chromosomes until they are lined up in the center of the cell. This is a critical safety check. If a chromosome is not perfectly aligned and attached to both sides of the spindle, the cell cannot guarantee that the DNA will be distributed evenly in the next step, so division will pause. Anaphase is the stage where physical separation occurs. The molecular glue holding the matching chromosome halves together is broken, and the fibers pull them towards opposite ends of the cell. By the end of this step, the cell has two identical sets of DNA positioned at opposite poles, ready to become separate nuclei. Telophase and cytokinesis often go hand in hand. In telophase, the cell returns to its normal state. New nuclear membranes will form around the DNA, and then the chromosomes relax. Cytokinesis then completes the process by splitting the cytoplasm. In animal cells, the membrane pinches inward to divide, but plant cells must build a new wall down the center to separate the two new cells. This marks the end of the meiotic phase and marks the beginning of a new cell cycle, which starts with the interphase. Since DNA contains the instructions for life, moving it is a very sensitive task. If a cell receives too much or too little DNA, it will fail to function properly. To prevent these errors, cells will use checkpoints that act like traffic lights, ensuring everything is correct before moving on to the next stage of the cycle. These checkpoints are essential stop and go points. The three main checkpoints occur at G1, G2, and M. At each point, the cell evaluates its health, DNA integrity, and environmental conditions. If any problem is found, the cell stops the cycle to perform repairs, and if the damage is too severe, the cycle will not continue. The G1 checkpoint is the point of no return, where the cell commits to division. The cell checks for sufficient size, nutrients, growth signals, and DNA health. If the cell does not receive a go signal, it exits the cycle and enters a resting state called the G0, which was mentioned before, where it remains alive and functional, but it no longer divides. In case of severe damage, the cell may instead be directed towards apoptosis, where it kills itself. The G2 and M checkpoints are very close together. The G2 checkpoint ensures that DNA was copied perfectly during the S phase. Later, the M checkpoint occurs during metaphase and monitors the tension on the spindle fibers that hold the DNA together. The cell will not proceed to anaphase until it is certain that every chromosome is properly attached and ready to be pulled, preventing unequal DNA distribution. The engine that drives the system is made of two types of proteins, cyclins and CDKs, which are the abbreviated forms of cyclin-dependent kinases. CDKs are always present in the cell, but remain inactive until they bind with the cyclin. Because cyclin levels rise and fall in waves throughout the cycle, as seen both here and here, they act as this timing mechanism that controls when the cell can pass checkpoints. A key regulatory protein is p53, which is often called the guardian of the genome. It is mostly seen in the G1 checkpoint, where it checks for DNA damage. If it does detect damaged DNA, it halts the cycle to allow for repair. If the DNA cannot be fixed at all, it triggers apoptosis, or programmed cell death, which prevents the damaged cell from dividing and harming the organism later on. Another important part of this unit are feedback loops. The cell cycle itself uses feedback loops to maintain balance. Positive feedback loops push the cell cycle forward once conditions are met, ensuring that division proceeds efficiently. In a positive feedback loop, it often starts at equilibrium and then exponentially deviates away from the midpoint. Negative feedback loops, on the other hand, stop progression when errors are detected, where they start from a deviation from the equilibrium and move towards a midpoint. Together, these loops ensure that the process of cell division is both smooth and safe. But sometimes, even these extensive safety systems to regulate the cell cycle can fail. And when these safety systems fail, usually due to damage in the genes that regulate p53 or cyclins, cells begin to divide uncontrollably. This is the definition of cancer. Cancer cells ignore checkpoint signals and continue to multiply even without growth signals, eventually forming tumors that interfere with healthy tissue. Great, that's the cell cycle. Now let's move on to some questions. First, I will read the question to you, then you can pause the video and come back when you have an answer in mind. Let's start with question 1. The coagulation cascade controls blood clot formation in response to blood vessel injury. Thrombin is an enzyme that plays a key role in regulating the coagulation cascade. A simplified model of thrombin's role in regulating the coagulation cascade is represented in figure 1. AT is a competitive inhibitor of thrombin. Which of the following effects on the coagulation cascade is most likely to result from inhibiting thrombin activity with AT? A. The activation of clotting factors will be blocked. B. The rate of fibrin formation will decrease. C. Thrombin will be converted to prothrombin. Or D. The rate of blood clot formation will increase. Please pause the video here and come back when you have an answer. The correct answer is B. According to the diagram, thrombin converts fibrogenin to fibrin, which leads to blood clot formation in the first place. Inhibiting thrombin with AT would decrease the production of fibrin. If you didn't guess B or choose the answer choice B, I'm guessing that you picked the answer choice A. So why is answer choice A wrong? The activation of the clotting factors actually, will not actually be blocked, because what's fueling it are the blood vessel injuries and not thrombin itself. Blocking thrombin primarily slows the final step, but it doesn't necessarily slow all the upstream factors, and the blood clot formation will still proceed. Here's question 2. After fertilization, multicellular organisms undergo the process called cleavage, a series of mitotic divisions where the egg's large volume of cytoplasm is divided into numerous smaller nucleated cells. An irregular process known as direct cleavage occurs when one blastomere, a cleavage stage cell, divides directly into three or more daughter blastomeres. Explain why the daughter cells are less likely to be able to successfully develop and become implanted as a result of this process. Please pause the video here and come back when you have an answer. Here's what your answer should look like. Direct cleavage produces daughter cells with unequal amounts of cytoplasm and DNA distribution, which in turn disrupts normal gene regulation and cell signaling which is required for development and further division. As a result of this, these cells are less likely to properly develop, which reduces the chances of successful implantation. Here are the key takeaways from this video. In summary, the cell cycle is a highly regulated process. Interphase handles preparation for division, mitosis ensures even DNA distribution, and checkpoints protect against errors. You should remember the steps of mitosis using PMAT: prophase, metaphase, anaphase, and telophase, followed by cytokinesis after. States like G0 and processes like apoptosis prevent damaged cells from dividing. When regulation fails, it can lead to cancer, which is why understanding these control points is a central focus in biology. That's the cell cycle. Controlled, precise, and absolutely essential. Thanks for watching, and I'll see you in the next video.